Welcome to the Global Addiction Therapeutics Innovation Day.
Professor Antonio Verdejo-Garcia. So we are very happy to have Antonio here. He flew from Melbourne to here and arrived just a few minutes ago here and was able to make it to University of Hamburg. Antonio is a full professor of psychology, University of Monash. And just to give you a kind of an overview about what he's doing, he's one of the world-renowned figures of addiction psychology. My psychology students, when they want to talk about somebody who they really like to be like him or her, they usually say, we really like to be like Antonio in the future. So he's a role model around the world for psychologists to be really influential in the field of addiction and addiction psychiatry, addiction neuroscience. So he has been very influential in developing lots of neuroscience findings, lots of psychology, lots of treatment developments, and he's going to enlighten us about fantastic things that he's doing in Monash University. Antonio, please.
Thank you so much for the introduction. I literally arrived like 30 minutes ago, so I've done many crazy things in my career. I think this is probably the craziest one. So, but I really appreciate the invitation and really appreciate the flexibility for fitting me in, because obviously I missed my spot before. So thank you very much for that. So yeah, we'll be brief. I'll be chatting about like the work that we're doing to try to use brain imaging to inform the brain stimulation interventions for people with alcohol use disorder. And we're focusing on a phenomenon that is very well known by all of you, which is craving. So like I've always found this painting, this artwork to be very suggestive of the experience of craving. You probably recognize that is Ulysses tying himself up just to prevent himself from falling into the charms of the sirens. And it kind of gets you a little bit of the gist of what the craving experience is, right? Like on the one hand, it's incredibly overwhelming, and at the same time is quite multifaceted or diverse, right? Like it has the motivational appeal of feeling the temptation. It has the sensory element of feeling, in this case, the song of the sirens. In the case of craving, we feel all the body tuning into the craving experience. And it has as well the element of conflict and desire for control and strive for control that sometimes one can actually exert, and some other times you can't, right? So we've been very interested in anchoring part of our neuroimaging research around this concept. And actually, when you look at the brain, and this is a render from some work that I did with Mark and others, in this case, what I'm trying to illustrate here are the different regions and systems that are involved in the craving experience, right? And you have a multiplicity of them, again, like you have from the ventral and dorsal striatum, which are more involved in the motivational and the habitual aspects of the craving experience, to the insula that is more related to the interoceptive experience, to areas like the ventromedial prefrontal cortex and the dorsolateral prefrontal cortex that somehow provide like online representations of reward value and also of goals to be able to manage that experience and align it with decision making and behavior. And the interesting thing is that these days, like you can trace network communication or neural communication within these brain regions, and you can better understand like what is the nature and what is the directionality of the connections within different networks. And in our case, we were very interested in what I'm going to coin today here as the craving network. I don't think it's a very standard concept, but it kind of encompasses like these different regions that I've been discussing so far. And so what we've been doing is we've been using this technique that leverages resting state data to better understand the valence and the direction of connections within a network. So essentially, it's going to tell you whether the connections within that network are excitatory or inhibitory, and it's going to tell you like where is the connection coming from and where is it going to. And therefore, it gives you kind of a very comprehensive picture of what is occurring within a network. And it is better to constrain it a little bit because if you try to analyze the whole brain that way, you can get mad. So I think it is way better when you have a network of interest like the one that I was defining before, and then you try to analyze like how those connections are operating. So the first thing we did, because let's say the long game that we have is to try to leverage this technique to understand what's going on for every individual in that network, and then to be able to personalize or to match brain stimulation interventions to the way the network is altered in that particular individual, right? But that is still a bit far. Like that's still something that I haven't been able to achieve this year, but it's the long game. The first thing that we wanted to do is to see whether that network was actually clinically relevant. So if we analyze that network on resting state data and we compare people with alcohol use disorder and healthy controls, like do we see some differences? And we saw some differences. Like I will go more in detail into these differences in one of the talks that I have in the next few days. But effectively, you can see a number of alterations in the way excitatory connections are altered and also the way inhibitory connections are altered in people with alcohol use disorder versus controls. And this is coming from resting state data from Enigma Addiction. So you have here about 452, well actually 452 people with alcohol use disorder and 239 controls. And this is the type of matrix that we generate. I'm going to show you a few of these, and in the next few slides I'll try to explain a little bit more. But effectively is a Bayesian estimate of what connections are impaired and what is the directionality and the valence of that connection. And this is the long game, as I said. Like many of you, like I think I know who I'm talking to. Like many of you clinically, like you would have observed consistently that no patient looks the same, right? Like they all have very different clinical manifestations. And for a very long time, we have had that intuition that that must be related to some type of heterogeneity in brain underpinnings that we should be able to capture at some point, right? And so far we've been unsuccessful, and we continue to be unsuccessful, but that is the idea. So the idea is that you have a network like the one that I was defining with 7 different regions. But the way those regions communicate in each individual is going to be different. And the way that communication is altered in different individuals is going to be also different. So whether that can give us a window into how we have to interface with the brain and with behavior when we treat people with addiction, in this case, people with alcohol use disorder, that is what we are interested in. So we got funded to test this idea, and in principle we thought that we would at least be able to identify like 3 different subtypes, and those are the ones that you can see there in the image. So this is a model, this is not real data, this is kind of like some of the assumptions that we had. And then what we thought is that if we identify like those 3 subtypes, one that is maybe more associated with alterations at the level of the ventromedial prefrontal cortex, another one more at the level of the executive control system, so the dorsolateral prefrontal cortex, and another one more at the level of the insula, then we should be able to apply brain stimulation interventions that are matched to those subtypes. And that is what we are trying to do. The interesting thing about this, and it was so exciting to hear like all the presentations before with the innovations, and I know there is an industry session right afterwards, is that if we manage to do this, like we will be leveraging a resting state sequence. That's everything we use. So we use 7 minutes of resting state to extract all of this information about how the network is impaired in a particular individual. And that has very interesting implications for how to apply it in real life, right? Because one of the things people always say is like, why would you do brain imaging? It's very expensive. You're never going to be applying it clinically. Well, this would be a way of applying brain imaging clinically in a very realistic and tangible way, right? Are we there? No, we're not. But we're progressing towards it. So what we've been doing, like we got funded for this project literally 1 year ago. So we've been working into this for less than a year. So everything I'm going to show you is very provisional, but I was very excited to show it to you, and my intention, and that's why I rushed into the into the conversation today, is to continue the conversations in the next few days, right? But what we've been doing is, on the one hand, we've been leveraging the resting state data from Enigma Addiction, and as you saw, like there's more than 700 participants in that data set. And in parallel, we are collecting our own data, and we're collecting it in this facility, which is called Brain Park, which is a very interesting facility. It's located in our university, and it collocates the brain imaging center with the clinical suites. So effectively, you can be imaging people, and then you can be bringing them to the clinic to do whatever you want to do, whether it's psychotherapy, or it's neurofeedback, or it's psilocybin. We're going to be doing a few things, but I need to focus now. So for now, what we've been doing is, by leveraging the data both from Enigma Addiction and the resting state data, we've been applying like unsupervised machine learning algorithms. So in this case, this modeling that is called variational Bayesian Gaussian mixture modeling. And we've been including as training features the connectivity between those 7 regions that I mentioned before. So effectively, like, is connectivity within the craving network. We are analyzing both like average metrics and distributions. So you see there 2 different parameters. So one is an estimate of the mean connectivity, and the other one is an estimate of the variation within that connectivity. We ran into some problems with the data, because for this type of analysis you require a certain quality of the time series, so we had to filter some of the Enigma data, and we ended up with a sample of 240 participants, and that includes 60 that we have collected this year, plus the ones that were available for Enigma Addiction. And the first thing that we were asking the data is, are you classable, or are you better interpreted as something homogeneous? And the data was telling us that it's classable, so you can actually distill heterogeneity in the data. And then the second question is, okay, if you're classable, how many classes are the optimal solution? And we thought it would be 3, but all the data so far seem to be pointing to 2. So far, like, we've been able to derive 2 different subtypes. They are very consistent across sequences, so you can see them here, like across 2 consecutive sequences of resting state. And they also very consistent across sites. So when we analyze, like, Enigma Addiction is composed of many different sites. There's some people here that are contributors to that data. So when you analyze the classification across different sites, like, it's also very consistent. So there's a few indices of what is the optimal number of clusters. This is one of them, the silhouette scores, but we've been using some others, just basically to analyze the fitness and the stability of the clusters, and that seems to be the case. For now, like, 2 seem to be the result that is more plausible. And then here, you can see these are, so this is, why? I think I missed a slide, like, because there is a subtype 0, but I cannot find it here. That's probably because I sent the wrong slide. But anyway, don't worry too much. I'll show you how the PEP matrices work, and then there is a slide that summarizes. And as I said, like, these are still very preliminary results. But effectively, what you can see in here, in the left side, is the mean connectivity measured with spectral dynamic causal modeling, and you can see here where the connectivity is coming from. So, for example, in this case, from the left dorsolateral prefrontal cortex into the ventral striatum, or from the left dorsolateral prefrontal cortex to the dorsal striatum. And when you see the boxes in blue, that's because it's an inhibitory connection. And when you see the boxes in red, it's because it's an excitatory connection, right? So here you can see when you analyze the matrix, and this is for one of the subtypes that we have obtained, and this matrix combines both the subtype and controls. You can see what is the valence of the connections and where the connections are coming from and going to. Then, like in the second matrix, what you can see is what are the differences in terms of connectivity between the subtype and the healthy comparison group. For example, this subtype is basically characterized by alterations stemming from and coming to the dorsal striatum, which was an interesting thing. It was not part of the original hypothesis. So here you can see a summary of what the subtypes look like so far. What we have is one of the subtypes that actually is the one that happens in the majority of the participants that we've been analyzing, that is mostly characterized by alterations in top-down modulatory control, mostly stemming from the ventromedial prefrontal cortex and mostly related to both reduced inhibition and reduced excitation. And then another subtype, which is the one that I was showing, the matrices, that is more characterized by alterations in subcortical connectivity or in connectivity that is stemming from subcortical structures. We're still revising and revisiting these results. So now what we have done is reprocessing all the Enigma addiction data to be able to reanalyze the results, not in the 200 and something, but in the 700 and something that we have. And we're in the process of redoing all that analysis pipeline. So we'll see if these results hold up, but so far they've been pretty consistent across the different iterations of the analysis. What we've been doing in parallel, and again, this is very aligned with the innovations that I've been hearing about before, this is about increasing our computational power and our ability to make rapid decisions. So we've been designing this subtyping pipeline that effectively what it does is it streamlines the neuroimaging analysis for every individual to a level that you can welcome a new participant, do the brain scan, and in less than 4 hours, you have all the pipeline completed, and you can get an outcome of whether they belong to one of the subtypes. And then you can allocate one of the interventions that you think is optimal for that subtype. So this has been running in parallel. Another part of the team that has not been involved in the analysis have been able in creating this pipeline to be able to, in a very computationally efficient way and time-efficient way, being able to subtype individuals as they come. So what we're going to be doing is, as soon as we are confident with the subtyping, we're going to be bringing people in, imaging, subtyping, and inviting them for an intervention on the next day. So it's a way of making things as quick as possible, as quick and efficient as possible. Again, we're going to be doing that in Brain Park, and we're going to be testing different, well, let's say that it's still to be decided, what are the matching brain stimulation interventions that we're going to be applying, and that's something that I am hoping to discuss as well during these days. The team has been working with the BrainsWay system, so with the wide volume TMS or deep TMS, and we have validated a few protocols that we thought would be useful for some of the subtypes. We didn't get everything right, but we validated a few. And we keep playing with the different strength of the coils for the different regions of interest. So again, interested in hearing your thoughts. But effectively what we want to do is, one, we refine all the subtyping pipeline to being able to select what is the potentially best brain stimulation intervention for each of the subtypes or biotypes. So this has been possible thanks to, I would say, a quite transforming grant by the Wellcome Leap And I would like to publicly acknowledge them for being, I think they've been brave, we've been brave as well. All of this work has been generated in approximately 9 months, so, and there's still two plus years to go, so it's been quite intense. These days I sleep on planes, like not at home, but it's been great. And thank you very much to the team. Like this is an exceptional team, including some people that is very well connected with ISAM, like Arsha and some of the people that originally developed spectral DCM, which is the technique that we're exploiting, especially Adil Ratzi. And I would like to publicly acknowledge everybody that has contributed to Enigma Addiction. Like, thanks to that data set, we are doing this analysis and we're moving forward with this project at a speed that I didn't think it was possible. So thank you very much to everybody that is generous enough to give their data for other people to use it. Thanks a lot.
Thank you very much, Antonio. Fantastic presentation. And those who do not know Enigma Addiction is a global consortium on addiction neuroimaging. So multiple different labs, they are developing their brain imaging database and they are sharing the databases together. And we have different divisions within addiction, Enigma Addiction. We have resting state, we have DTI, we have fMRI drug cure activity, so there are different sub-consortium within the Enigma Addiction. And if you are interested to join, I will be happy to provide you with further details. During the first day of the conference, we have 2 panels on neuroimaging, informed interventions, and neuroimaging, how neuroimaging is going to reshape the future of addiction medicine. You can definitely join us and we'll discuss about those specific collaboration opportunities. So we have time for 1 to 2 questions. If there is any question in the room, otherwise you can ask questions. Okay. Yes, sir, please. Can you just...
Thank you so much. So it's a way for more precision medicine. I want to ask you, are the cluster you identify, are they stable or not? Or what do you know about their stability? Thank you.
So the question is if they are stable, right? Yeah. So all the stability diagnoses that we have run tell us that they are stable. The other thing that was encouraging is that we've been able to replicate them in 2 sequences that we acquired. So like what we do is we do a resting state sequence, then we do some more stuff, and then we do another resting state sequence, and we've been able to replicate them across the 2. And the other thing is that we've been able to also see that the distribution of people that is classified into each of the clusters is quite consistent across different sites. So if you take Enigma Addiction, you would have like maybe 13 different sites that have contributed data. So when we apply the analysis, like the same kind of like breakup seems to emerge in every single site. So it is encouraging, but still it's provisional, right? Like we're still working on this, and I think like probably in the next month or so, we will probably have like more definitive results. But so far it's looking stable by all the diagnoses that we've been able to run.
Just a quick question. I'm Kathleen Brady, wondering if you know anything yet about the clinical correlates of those 2 types, because there's this old typology back to the 70s, Cloninger, with alcohol dependence.
Yeah, thank you very much, Kathleen. That's a really good question. I think we're still in the infancy of this. Like, what we have seen so far is that one of the subtypes has way more intense cravings than the other one. And I'm sorry, but I can't remember which one is which. And then like so the thing that we're very interested in, so in parallel to this, we've been doing a very comprehensive cognitive phenotyping focused on reward processing, executive control, and interoception. Then what we're doing now is we are deriving metrics through computational modeling, and we are embedding those metrics into the imaging matrices. So rather than just looking into how the subtypes do behaviorally, like we're embedding them as a third covariate into the imaging matrices. So we have to do this process one by one, and we have to decide which are the ones that we think are more promising to be able to tease them apart. So that is why it's still work in progress, but yeah, that is the expectation, that they're gonna differ on some of those cognitive phenotypes in a way that it could be nicely integrated with the biotypes.
Yeah, if you'll remember way back to the old typology, genetic predisposition was very clear for type 1 and type A.
That's right. Yeah.
So it'd be interesting to look at that.
Yeah, absolutely. Thank you.
Organized by International Society of Addiction Medicine, sponsors and co-organizers CNRE, Indivir, Spark Biomedical, Salofa, Ethifarm, University of Minnesota, European Union Drugs Agency, UK Office for Life Sciences,